Samar Tarazi, a PhD student from Palestine. I work with the Institute of Biological Information. It is the Institute of Mechanobiologie, and we are looking for the neurons under mechanical strain. Normally, there is a tissue deformation in brain, and this happens during brain development or during daily body motion. And more especially and interestingly, that this happens within every heartbeat. And this is illustrated here in this movie from Term and his colleagues in 2018. Also, other extreme deformations happens during car accidents or with athletes, especially with football players. Or other deformations happens with cancer tumors in brain, and these are static strains. But usually studies are looking for the effect of chemicals and molecules on neurons, but they are lacking studies in the mechanical effects on neuron behaviors. And in this study, I'm focusing on neurons from rat corticals. They are embryos at day 18, and after this isolation, we are seeding these cells on an environment mimicking the physiological environment in vivo. They are elastomeric stretchable 50 kilopascal substrates, and we are applying after that a mechanical strain using a homemade uniaxial stretching machine. With these setups, my colleague Yella Abraham in 2018, she published an article showing that neuronal cyclic stretching has an effect on formation retraction bulbs and collapsed microtubule networks, and on the long term it shows a reorientation perpendicular to the strain direction. But however, this cyclic stretching didn't show any sign of apoptosis, so neurons adapt to cyclic stretch. Moreover, in this publication, it showed that, and in other publication too, it shows that neurons have a higher tendency to make more branches and to have longer axons. And this is shown in the first image here with a light green color showing more branches and new branches. And it can be shown in the first upper graph here how the number of new axons or new branches are enhancing. So we can see that the number of branches in the unstretched neurons is between 25 to 30 branches per neuron, while for stretched samples we can see that they are from 30 to 35 branch per neurons. And also in the lower graph here, we can see that neurons are longer when they are stretched cyclically. From this study and other studies, they are always looking for the effect on the structural components of the neurons, but they are not looking for the functionality of these neurons. So in my study, I'm trying to see if these neurons are still active during this different mechanical strain, if this cyclic strain and enhancement in branches and elongating this axon is enhancing the communication between cells. And the other question, and the most important, is how much do these neurons tolerate the different amplitudes of stretching? But before going deep inside my experiments, first we want to know more about neurons' activity. and they are usually described as a rapid change in the membrane voltage, and this is coupled with a calcium ion concentration change in the cell, and this is producing action potentials. These action potentials usually go in only one direction, from dendrites and into axons, and from one cell to the other. In my experiment, to label these calcium potentials, I used a fluorescent dye called Cal 590, and I start recording calcium ions only at day 9 of seeding the cells. And this was the best day because, and in my experiments, I found that the more cells, the more activity, the more days, the more they are active. And to see the effect on and the behavior on the single neuron cell level, I used the concentration of 350,000 cells per chamber. While to see on the network behavior level, I used a higher concentration of 550,000 cells per chamber. My main experiments are mainly four. The first one is stretching during the network formation, and this mimics the normal change in and deformation in the brain that happens with breathing, with body motion, or with heart beating, and it is 15% and 300 millihertz. Here is the video recording of calcium ion changes. The first video here, we can see the unstretched control. It is on the network level, so here we are using the high concentration cells. They are highly active. They are showing that most of the cells are active and synchronizing at the same time. While when we are comparing this with the second video here, after 48 hours of stretching during the network formation, we cannot see any difference. Most of the cells are active and all of them are synchronizing together at the same time. Even if we are stretching for the whole time from day 1 and until day 9, there is no detected effect on the activity of the neurons. While going down to the single neurons level, the activity in the unstretched controls are only random and a few active cells. They are not showing any synchronized activity together. And it is the same with 48-hour stretch and the full-time 192-hour stretch. But we can see clearly how these cells, after being stretched continuously, they are clearly reoriented perpendicular to the stretch direction. And to have a closer result with number or quantified results, Dr. Shar and Giyok helped me to develop an algorithm, and it is mainly took the most time to do a segmentation for each cell in the video and to find each signal and its intensity and the period of the signals. And from the first graph here, we can see that around 90% of the whole cells are active and around 80% of the cells are synchronizing together, and from around 5 to 10 synchronizing peaks are per minute, and this frequency and the synchronized cell number and the active cell number are all without any significant difference between unstretched and stretched. The same was done for the single neuron levels. We can see that there is a very lower compared to the neurons network level. They are only 20% of the cells are active, and only 10 to 20% of these cells are synchronizing, or sometimes nearly zero. And there is from 0 to maximum 1 peaks per minute. and there is no differences between unstretched or stretched. And from these results, we can conclude that enhancement in cell length or enhancement in branches do not enhance the cell-to-cell communication in a network, but they are mainly just for stabilizing these cells on the area. And also, cyclic stretching with only 15% amplitude is not making the cells losing their activity, so they are robust and resistant to these amplitudes. My next experiment was to increase the amplitude, so I used 20% and 30% for 300 millihertz, and we wanted to see if there is an effect on the activity, if they can tolerate this high amplitude. And from the results here, we can see that the unstretched, the 20% stretched, and the 30% stretched samples are all of them highly active, and they are not affected with these stretching amplitudes, even if we looked for recordings after 24 hours. And we can see this, that there is no significant differences in the graphs, only there is a slight difference in the number of active cells with 30% amplitude. And the third experiment was trying to mimic the tumors inside brain. So tumors are statically stretching the neurons with a very low speed. So I tried stretching 30 and 60% in a very slow speed of 0.05 millimeter per second. Also, the calcium ion changes are stable with 30% stretching, and even after 72 hours of relaxing them, while there is a significant and high significant effect if they are statically stretched for 60%. So we can here conclude that neurons and networks are stable and resistant for many amplitudes, but until 60%. My last experiment was to mimic the brain injuries, like a car accident or whatever. So I tried 30% and 60% with a very high speed of 80 millimeters per second. And the results are shown nearly the same as the static stretching experiment. So the 30% is stable activity, while the activity is nearly lost with 60% stretch. But what is interesting in this result, that, and also in the static stretching results, that after 70 or 24 hours and also for 72 hours after these accidents, the activity is regained as normal and as hyperactive after 24 hours. So we can say that neurons can adapt after 24 hours for these high amplitudes. And from these results, many questions has arisen. We want to go deep inside the molecular effects. So we wanted to ask, does there is a more or less functional synapses with stretching amplitudes? And to see these active synapses, we tried using fluorescent markers. The first one is synaptophysians, shown here in red color, and the second one is PSD95, a postsynaptic marker, shown in green color. And the colocalization, shown in yellow color, shows where there is active synapses. And to have these results and quantified results, Georg and Ronald helped me to develop an algorithm, and the results showed that there is no significant difference between stretched or unstretched, even in the 60%. So the previous result was completely different than the videos and recording showed before, and this made us think about what happens inside neurons. Are they stressed or not? And that's why we tried the autophagy test, and we tried it with the LC3B marker. It is a stress marker, and surprisingly, these neurons do not stop surprising us. The results showed that only the cycling stretching of 15% showing a high significant difference and increase in the stress markers, while the 60% amplitude didn't show any significance in stress markers. And to go more deep, we tried to see the inflammation markers, the interleukin-6 expression in cyclically stretched neurons, and they were increased by 2 to 3 folds. But going back to the literature, 2 to 3 folds are not recognized as a significant difference and increase, or it is not even an inflammatory expression. So to conclude and to summarize all of these different experiments, I can conclude them to say that brain cyclic stretching do not enhance cell-to-cell communication, but it is only stabilizing the cell. And also that neuron networks, when they are formed, they are very stable and they can adapt to many different stretching amplitudes until 60%. And the most interesting point that after 60%, if they are relaxed for 24 hours, they can regain their complete activity again. And finally, I could say that neurons adapt to different mechanical conditions without any sign of apoptosis or inflammation. But many different future plans we are making to go deep inside molecular testing of these amplitudes and to try to culture these neurons in mixed culture, especially with astrocytes. Thank you very much for taking the time listening to my presentation today. I also want to thank my colleagues in IBI-2. I want to thank my supervisor in Germany, Dr. Bernd Hoffman, and my supervisor in Palestine, Dr. Firas Al-Batta. And also I want to thank the PGSB for giving me this opportunity. Here is my contact information. Thank you.